human development requires millions of cells to divide, migrate, and assemble into complex three-dimensional structures within a matter of days. To understand how the body's primary axis forms, we have to map the exact physical transformation of a transient microscopic structure known as the notochord. We will use these detailed handwritten clinical notes as our blueprint to decode the sequence. This 3D model illustrates our starting point, the flat, trilaminar embryonic disc. It consists of three distinct cellular layers stacked on top of one another: the ectoderm, the mesoderm in the middle, and the endoderm on the bottom. To orient ourselves, we define the longitudinal axis. One end is cranial, pointing toward the future head. The opposite end is caudal, pointing toward the tail. At the caudal end of the disc, a linear band of proliferating cells emerges. Far across the disc at the cranial end sits the prochordal plate, which marks the site of the future mouth. If we move along the primitive streak to its uppermost cranial tip, the cells gather into a concentrated, raised thickening. This specific structure is called the primitive node, or Henson's node. As the notes classify it, the primitive node is known as the organizer, as it regulates important processes such as right and left side determination. This node serves as the launch point. We will now track the continuous cellular migration out of this node to map the strict four-stage topological transformation of the notochord. Mapping this spatial sequence is the strict prerequisite for understanding how the entire central nervous system is subsequently built. All the critical activity begins inside the primitive node itself. Looking at a longitudinal cross-section, cells multiply and drive forward in a straight line toward the prochordal plate. Notice the spatial plane. Cells are sandwiched strictly within the middle mesoderm layer, navigating between the ectoderm and endoderm. This tightly packed cylinder is the notochordal process. The clinical notes confirm this phase, describing how these multiplying cells form a solid cord called the notochordal process, or head process. Back in our cross-section, the blastopore hollows out the solid head process. The rod becomes a tube, the notochordal canal. Immediately, its floor dissolves. This creates a temporary opening, allowing fluids to flow from the amniotic cavity down into the yolk sac. The text documents this exact sequence, noting how the whole canal eventually comes to communicate with yolk sac. This happens because the early embryo lacks a cardiovascular system. It requires a temporary nutritional shunt to feed the rapidly developing nervous system. The early notochord is an active, necessary life support conduit, supplying the materials required to sustain the embryo before the heart begins to beat. Shifting 90 degrees to a transverse cross-section, we observe the next shift. The hollow canal's walls unroll and spread outward. As they flatten, these cells seamlessly integrate into the roof of the underlying endoderm layer. This transient, flattened sheet is the notochordal plate. We can verify this counterintuitive phase in the notes, where it states the canal becomes a flat plate of cells called the notochordal plate. Suddenly, the process reverses. The flat plate's edges curve downwards, curling inward to form a tight U-shape. The cells proliferate until the edges meet, pinching off from the endoderm and rising into the middle mesodermal space. This fully detached cylinder is the definitive notochord. Again, the source material maps this exactly. The tube converts into a solid rod of cells. This rod is the definitive notochord. It gets separated from the endoderm. As the surrounding embryo enlarges, this newly isolated notochord elongates down the midline. This sequence of unrolling, integrating, and refolding provides the geometric mechanism required to position the structural axis completely separate from the digestive gut tube. The definitive notochord now resides stably in the exact midline of the embryonic disc. In this cross-section, the glowing gradient represents the notochord executing its most vital function, transmitting chemical signals upward to induce the ectoderm. Triggered by these specific chemical cues, the overlying ectoderm reacts by physically thickening into a dense band known as the neural plate. That neural plate then folds inward on itself, eventually pinching off to form the hollow neural tube, the anatomical precursor to the human brain and spinal cord. This induction pathway is confirmed in our text, which states the germ discs start differentiating into neural plate and neural tube. If we move the developmental timeline forward, the final fate of the notochord becomes clear as the human skeleton begins to form. There is a common misconception that the notochord turns into the spine. It does not become the vertebral column. Instead, as the permanent bony vertebrae construct themselves in a column around it, the notochord undergoes widespread apoptosis, largely disappearing from the body. This diagram of an adult spine shows its primary remnant, the nucleus pulposus, the gelatinous core that cushions the center of our intervertebral discs. If we move to the top of the cervical spine, we find its secondary remnant, a specific ligament attached to the axis vertebra near the skull. These exact remnants are thoroughly documented in the final lines of our clinical notes, explicitly identifying the nucleus pulposus and the apical ligament of Denise of axis vertebra. The notochord serves as a transient, microscopic scaffold that chemically programs the entire human nervous system, only to ghost away, leaving just a trace of its existence in our spines.